also without the computation or mathematics. And first we get the experimental data in our laboratory and make a, for example, mechanistic model made of the molecular interaction of the, the focus of interest. And this is an example of the cancer signaling network started from the AB receptor signaling, which is a receptor which is very important for cancer progression. And we make a model of this network and then perform the simulation based on the data and generate a new hypothesis. Then we validated those hypotheses using our experimental technique again. So we learn, the cycle this procedures as much as fast as possible. So, and then this is an example what we did so far. This is work we did in Riken actually. So as I said, this is the example of the cancer receptors of the RB receptors, which binds to the those external ligands, which are usually in the blood, and then they come to the cell surface and binds to the receptor, and then they have an interaction with this red-colored molecules, and this is a kind of a cascade, the marathon relay, so that they transfer the information of this interaction to this kinase activity, and then the other downstream kinase activity, and then the information coming from the outside of the cell are transmitted to the proteins, which is called the transcription factor in the cell, and then the transcription factor activate bind to the DNA in the nucleus, then the produce a new mRNA synthesis. So because of this complexity, but also redundant interaction from the same receptor, and we wondered how that the cancer cell decide which this cell need to be proliferated or another cell need to be differentiated. That was the question of this study. So we also found that there is a difference in the duration of the ARC activity that is here. And then we want to know what happened to this C-phos transcription factor after in the condition of this sustained or transient ARC activity. So this is it's a complicated figure, but it shows that the ARC activity, MEK activity, MEK is upstream of the ARC, activate ARC, and then eventually produce C-phos mRNA and C-phos protein, something like that. And we also get this time course data. We obtain this time course data by the protein phosphorylation using a Western blot technique. It's very, very manual. And, at up to 90 minutes, just up to 90 minutes after the ligand, the growth factor stimulation. And as you see that this is called hereditary, it shows a differentiation of the cell, shows this kind of sustained activity, but at the end of the C-phos activation, it shows a full activation. But in the case of the EGF, which originally shows the sustained activity of the MEK and ARC, showed nothing in the C-phos activity. That is very interesting because such analogous information finally made a digital decision, full activation or non-activation. So we did I'm explaining just a scheme how we do, so that we did like a sensitivity analysis that which pathway or which interaction is most effective in output. In our case, the C-phos. And then the perturbator system, and they see that this sensitivity. So, and then as a result, we see that this, the higher big change was observed in this, which is actually the phosphatase of the ARC. Actually, it's the down-regulate the ARC activity. That is the most effective target to change this C-force mRNA and the protein. And then we perform the simulation of the knockdown. Knockdown is just kills this disc, and also the to see its effect on the C-force mRNA, and then in the EGF and the Hergelin condition. And actually, you can see that the difference, how that this disc knockdown affect this C-force in the EGF and the Hergelin. And we did also the experiments, but the simulation shows this kind of the sustained results, but there is experimental show that it downregulated. So that but the EGF shows a good match. So that we saw that there is something for the Hergelin regulation, only for Hergelin regulation. And it's a complicated story, but we found that there is another regulator, additional regulator, only in Hergelin condition, which replace C-force mRNA synthesis. And actually, we validated this with another experiment, and we proved that there is this kind of negative regulator, actually, which is produced by C-force, but in turn suppress C-force. So that we did many experiments to prove this. And then finally we got this kind of network structure, how the ARC in the cytosol control the C-force in the nucleus. And it contains this feed-forward loop in the C-force and actually for DISP. And also there is an AND gate that which shows a sustained ARC activity is important for the C-force protein production. And also there is a negative feedback from DISP to ARC, because we could see the effect of the DISP knockdown for the this C-force. And also by the simulation, we found another regulator, unknown F factor, to suppress C-force mRNA. So it's a really complicated structure, but once we identify this kind of structure, also we use the MCS7 cancer cell as an example, but we could explain another cell phenotype using this same structure and experiment and also the simulation. So we do our research like this. So in this way, we actually try to identify the mechanism of the signaling network, particularly in cancer. Because the cancer, this is a famous picture written by the Hanahan and Weinberg, and they show that there is almost 11 signaling pathway which receive the external signal from the blood or environment, and they activate the signaling pathway individually. And then they control the transcription or cell cycle or apoptosis. So it's like, how do I say, like truly, the signal transmission, like you use like a computer. The cell is like a machine actually. So that if we can find the rule, then we can manipulate and control the cancer cell to go into the cell death or more proliferation or something like that. And we actually studied many kinds of the signaling pathway, like RB receptor signaling pathway. That is, we spent a lot of time, but also the NF-κB immune signaling. But the NF-κB is also important for the inflammation. And we also studied the cell cycle or TGB. Recently, we published a paper on the TGF-beta pathway on the skin aging. So, and we wanted to integrate those the signaling pathway all together, like Julio does with his colleague. But it's really hard to integrate those signaling pathways. It's a lot, and we need the data. And also, what we can do, experimental can do is very much limited. So we want to rely on the, computational method more. So but we found that the problem that, the model, even in our group, each people use the different the different, simulation tools, different, description to make a model. So our student, Hiroaki Imoto, he is in Dublin as a postdoc. He proposed to make the modeling simulation tool. It's called Biomass, to make the experimental biologists easy to make a model by themselves. So that from this biomass, using this biomass, you can perform like a parameter estimation, data fitting to the experimental data of their own, and also sensitivity analysis to find the bottleneck for the further validation of the experiments. And actually, it's amazing that after the 4 years, almost this number of the downloaded we could find. It's a free software, so many people, it seems, they're using it. So the next question that we had is that it seems that the dynamics of the signaling network is very important, and we wondered if the dynamics features can be used for the reclassification of the cancer patient. And this is data shown from the American group of the breast cancer. So that the breast cancer is typically diagnosis for 4 types and treated in 4 ways. So there is luminal A, luminal B, and HER2 positive, and basal-like. So that it is the standard treatment for the breast cancer patient. But if you look at each patient, even in luminal A and luminal B, you have a different mutation for each patient and different copy numbers. So it means that classical classification is not good for, not good enough for the individual patient. And we saw that we maybe another way to, reclassify the patient using this dynamics. So actually, breast cancer has a lot of mutation or copy number variation in the signaling pathway. Actually, it's a lot. And this causes drug resistance to the normal chemotherapy. So we tried this problem. So but there is a challenges in the development of the patient model. So first we need to identify the network, and also the parameters, and also the initial values. So the network, actually there is several breast cancer model available in bio models or kind of cake, and we can modify it depending on that purpose. But initially we can make, and of course we make the model by ourselves. And also the initial value of the model, these days it's more easy because we can use the public database of the TCGA or CCLE. TCGA is actually clinical cancer sample database, so that they have like 30,000 patients or something like that. So that we can get the individual, the prognosis from the their data. And the CCLE is a cell line database. They have the transcriptome data and the drug response data. So we can use this, but this is for the transcript mRNA expression. But we can, convert this transcriptome data to proteome data by modeling. We can estimate the protein level from this transcriptome. The most problematic one is to define the parameter for each patient. So it's not actually easy because we cannot do, the patient sample is taken once, mostly taken once. You cannot allow the doctor take your sample many times. so that this data acquisition is impossible. So we need to think about another way. So what we thought is that learn the parameter, estimate the parameter from the cancer cell lines, because this parameter is usually interaction of the molecules. So the molecules interaction, such a parameter can be same in our body because it's molecule level. It may not be related to each patient. Of course, there is a problem of the mutation which affects such interaction, but I will explain later. So based on that idea, we obtained the time course data of the 4 breast cancer cells and trained the model of this time course and used the CCLE, the cell line database of the transcript level of these the cancer cells. These molecules, transcriptome of these molecules in the model as the initial value of the model. And then we got the common parameters from those models. In this case, we tried to get the 30 parameter, common parameter set from the simulations. And then next we got the parameters from here. So now we can use the real cancer patient data from TCGA as an initial value, and then run the simulation for each patient. And then we classify the dynamics based on this time based on the max value or this drop rate or AUC, area under the curve, for example. And then this is a kind of example. So we perform the patient simulation for the 377 breast cancer clinical samples. And we found that the max value is a good feature to classify. And this is the result. So this is the in silico simulation of the heroglobin and the EGF. And here is the max value of the AKT, ARC, and the C-force. And this yellow shows the prognosis, and this red and blue shows the subtypes of the breast cancer, which are recorded in a TCGA. And then we found that we could see a good classification, particularly for the red group. Red group is triple negative cancer. Usually they don't have any treatment method. They just get the chemotherapy and radiation therapy. They don't have any molecular target drug. So in this case, we found that even in a TNBC patient, there is a lower responder group to the heroglobin, which shows a more poor prognosis. And we also did the sensitivity analysis for each patient of this TNBC, and found that the poor prognosis patient tend to have a lower EGFR sensitivity, sorry to be small, but the lower the EGFR sensitivity and the better prognosis patient shows a higher EGFR sensitivity. And we use the CCLE, the public database, because they have a drug response data, and I'm cutting the very detailed skills here, but we validated that actually this, the beta prognosis group is actually sensitive to the EGF receptor targeting drug. So that we hope that it's opposite, and then we can show that that the poor prognosis group can be treated with the EGFR inhibitors, but wasn't like that. But at least from this analysis, we could see a kind of mechanism why that the TNBC patient there are two kinds of poor and better, and what's the reason of this prognosis. So the next things we are just doing right now and try to publish the paper is that the classification method, because in the last study we used the max value as a classification, but it's not that good because as you see we only use the 3 kinases as the features, but actually after the during the simulation we can get a lot of simulation data of the intermediate kinase and the protein-protein interaction. We have a lot of the in silico data which can be used for this classification. So we developed a deep learning method to classify the dynamics. And this is done by the Masato, who is a PhD student, but also he works for the company, and he tried to get a PhD in our laboratory. And he's very good at the AI. So he found that he compared several methods of this deep learning, which is most suitable for the classification of those dynamics. And then he developed the DIM profiler, which is self-supervised and non-constructive and label-free the learning. And he made this encoder and decoder system. And in this case, we used a lot of information of this simulation data, and finally showed it in 2D dimension. And he showed that what is the important feature, dynamic feature, to classify the patient. And this is the results. So that now it looks different from last one. So there is a luminal I and luminal B, and HER2, and treated TNBC here. So that we can see again that good separation of TNBC patient from the other breast cancer patient, but also this the survival prediction is much better than the earlier our own method. And from this analysis, we found that the kinase, which is called the GSK3 beta, can be a good marker for the high-risk TNBC patient. And he compared the transcript level of the GSK3 beta, because we can see this data from the TCGA data itself. But there is no significant difference in transcript level. But through this kind of dynamic simulation, we can see that this can be a potential biomarker to classify the TNB patient. And we also checked the literature, and in fact there are several papers supporting this hypothesis. And here is the next challenge. So that because this is about how to integrate this molecular dynamic simulation and the cell simulation. Because when I was in RIKEN, I closely collaborated with people who is involved in Fugaku or their former supercomputer project, who does molecular dynamic simulation. And at that time, we collaborated for the prediction of the protein-protein interaction. Predict the protein-protein interaction from the free energy changes before and after. So in our cell simulation, we could somehow develop the patient-specific model. But as I said, we can use the initial value of the model from those database, but we don't know how the mutation of each patient affect the prognosis. And then the mutation, such a interaction, mutation in protein, how they change the parameters of the system, which is very difficult, and nobody tried that. But doing this analysis, we can also integrate the parameters of the model from the MD simulation and the initial value of the model from the TCGA, and integrate those information in cell simulation. So I think it can be very, very challenging, but also very interesting. So in our breast cancer study, we noticed that this model was okay, even it's developed for the breast cancer, but we could classify the colon cancer and as ovarian cancer relatively well, but we couldn't do anything for the lung cancer. So we saw that there's a mechanism that as a reason why we couldn't do it. So lung cancer is well known for the mutation, EGFR mutation. So we tried how the mutation, that this is a typical mutation of the EGFR in lung cancer. So in this case, we used the 5 mutation and wild type. So we took again this kind of time course data of the EGFR and the SIC here, and MEC and ARC and C-force and so on. From it's complicated, but you can see this relatively, the dark color so comes top in here and around here, but only SIC. So we see this dark color comes lower, so that we saw that there might be an effect of this EGFR mutation affecting interaction between EGFR and the SIC. So then we try to integrate those the molecular level and the network level. So the network level simulation, we can perform the parameter estimation to identify the interaction of the K-off and the K-on of the EGFR and the SIC. And for the EGFR kinase itself, we can estimate the free energy changes before and after EGFR kinase and the SIC peptide. Unfortunately, we couldn't find a full-length SIC protein in this that when we started this project, but maybe we can use the structure given from AlphaFold now. That is what we are thinking right now. But the idea is that to compare that this this KD and the KD obtained from the MD simulation, and if there is a good correlation, then we can go integrate this information obtained from the EGFR to our cell simulation model. So we performed the MD simulation of this EGFR mutants and its interaction with the SIC peptide. And to estimate the delta G, we used the MMPBSM method. Actually, we used this method for several times. I already, there's people who does MD simulation, they say that it's not the best method, but they cannot find a more, better method. So that we use this method. And for this simulation, we use the Fugaku and also use the software. It's called Genesis, so that we can run parallel simulation of this MD on Fugaku. And then in case of the parameter simulation from the EGFR and the SIC, then we usually write the equation of the interaction of the EGFR and the SIC like this, and we can estimate this K-on and the K-off. And as I said, we can compare this large KD obtained from here and large KD obtained from the delta G. So this is what we obtained for the parameter experimental data for the parameter estimation for each mutant and for each molecules. So of course we need a lot of data here, but this is what we got so far because this is an initial try. So and also compare the delta G obtained here, and here is a correlation. So this is the x-axis is a prediction from the mass model, and the y-axis is a prediction from the MD simulation. So that the correlation is not that good, except if we don't have this mutation. But we are thinking to modify the model and also change the simulation method at the MD. We need to run MD more many times because we just tried 3 or 5 times per mutation, but for 2 microseconds. But now we are thinking to shorten the time and repeat more rounds. That is the next strategy. But this is what we are doing right now. So now I change the subject. So now I explain the patient-specific model, which we train the model using cell line data and add the patient data as the initial value and make that patient-specific model. And in this study, actually we developed a method. It's called text model. So so I will explain this one because this is actually became a good suggestion for us to let us know the next direction of our research. So text model, again, actually Imoto Hiroaki in Dublin now, he suddenly realized that this method. So that the mechanistic modeling, we usually use very simple equations like a mass action or Michaelis-Menten reaction or linear decay or something like that. And the description of the reaction is very simple. So that like degraded, translated, phosphorylated, or dephosphorylated, or activated or inhibited. Very simple. So he saw that maybe he can make a rule for the such modeling, so that at this stage, this is not like a natural language processing, but he used the pattern matching to correlate the, like if, for example, if we write that A binds to B, then automatically generate this kind of mass action reaction. Another is, the B phosphorylate A, then it's a Michaelis-Menten reaction, so that we can, this textile model generate the Michaelis-Menten reaction, and also we can generate like Hill equations and decay and so on. So from this simple rules, based on this simple rule, actually we could make this large model, which is consisted of the over 300 later equations and 200 species and 600 parameters. And this is not good only for the biologists to make a model by themselves, but it's good for the mathematicians, because when you're writing a code, when you find a bug, it's very difficult to find this, complicated equation. It looks all similar. So but by using this, you can find where is the mistake. So this is good for this such a purpose. So now we have like a text model and previously published biomass. And then, but still we have a problem, because to make a text model, to use some text model, we needed to know A binds to B. So to know A binds to B, we need to read the literature, many literatures. And then it takes time, and it's a manual creation mostly. So that you need a lot of time to reading papers and something like that. So that eventually I saw that looking at the Google Translate or DeepL, so that now the the language processing seems to be good enough to apply the method to biology, particularly for this kind of extraction of the knowledge from the literatures. So then we tried the use of the NLP for our method. The first thing we tried is not making a model structure from scratch. First thing is apply the already known such a structure, but by combining knowledge from the PubMed to make a cell-specific KEGG model. So the KEGG is very general, so that the KEGG written in here is not my KEGG. So we need to make our the model not myself, but also this disease-specific model. So we converted this Kiwamu. He is now doctor student, first year. He converted the KEGG to the KGML file to the other format. I forgot his format, but he format and converted to the text to model format. And then in other way, we he checked the co-occurrence of this each genes, and in this case the cell line name by checking co-occurrence of these molecules and each cell line name, we can make a cell-specific KEGG pathway and also perform the simulation like that. We can change it this name to like a disease name or the other name and test how the KEGG pathway is changed. We don't say that this is necessary to be incorporated to the this pathway, but this analysis clearly show that this MCS7 cells, this molecule is more important this cells, and this cells, this is more important than the MCS7 cells. So the next thing is that to make this kind of network from literature from scratch. And we tried many things. First is making this one-to-one interaction, like A and B interaction using PubMed, and we use the tool called PubData and also the pathway database. And the PubMed is used, PubData is used to extract the co-occurrence of the A and B, and Omnipath, the pathway database, it contains a regulation of these two genes. So by combining this, we can extract the co-occurrence of A and B, and then we can estimate how A regulates B or something like that. And we trained with this one with the BioBart, and then we estimated it's very small, but how A and B relationship is predicted. And we found that at least positive regulation or negative regulation accuracy is more than 90%. But from this analysis, we found that this method is valid if we care about just the A and B relationship, but cannot do more than 2. For example, 2, if it becomes 3, we cannot do. And if the A and B and the product B does something, then we cannot do, or we cannot predict from method. So we decided to make a LLM, to use a LLM. And this is our the first and very simple task we did. So that this is the paper of the mathematical modeling published a long time ago by the Boris Korolenko, and he described very details how to make easier to last pathway. So here is the description. So we asked GPT-3 to output the text model format, and we show them example. Please, give an output like this. And this is the results. So that this is a text model, and then here is black one is right prediction, and this orange one was the prediction that the GPT missed, and this gray one, sorry, not missed. They additionally predicted, which is not in the original paper. And the gray one was GPT missed reaction. And this is originally this is a modeling paper, so it's easy to validate the results. So we see that which is wrong and which is right. And the results tell me that if we start to make a model like a 1 month or modeling takes time. So but this level is just similar to the kind of beginners who make a model, but also it can be published if nobody believes that this is wrong or not. So and we also perform the simulation using biomass, and then we see that the original data is like this, simulation is like this, but the LLM-based model can also generate the more or less similar results. The quantitatively different, but the qualitatively satisfactory, I think. So the next what we are doing is to extend this method so that we just use one single paper to make a model, but it's not the case in the usual modeling procedure. So in this case, Kiwamu tried many things, and he decided to make a vector database from a PubMed, so that the information taken can be taken from the PubMed is only abstract. Abstract is made of 10 sentences or something like that, and we the process that such a sentence to the into the sentence transformer and make a vector database. And also we give input query that what we want. In our case, we want the EGFR signal pathway. And search the similar, the answer from the vector database, and also extract such information in a structured format, and combine those results to reconstruct a network. And this is also the initial trial that it's very much complicated, but we asked the GPT in our case, so that the after query is like this title of this the Boris Korodenko paper on the abstract of his paper. And we look at the 100 papers. Actually, it's on the Boris's paper as a references, and we could extract this EGFR interaction and the dimerization of the RB receptors and ARC and the other interaction, actually which correspond to this red colors in a cake. So that this is what has been done so far. But we think that this is not impossible to make a model this so much complicated one, and it seems very noisy. So that right now we try to use the improve the method by so that we reconstructed the vector database, the changing the window size and so on, and also we are try to use the log to improve the such a model to improve the query to the model. So that this is actually going on right now. And this is I'm sorry, take time. So this is our future plan. So I said, I am I'm a original biochemist, but I like modeling and I like the computational method, because I believe that the experimental method is very much limited. What we can do is very much limited. We even though the multi-omics data is given, but we can see the dynamics from the multi-omics, and even if you want, you need a lot of money to get the data. But on the other hand, what we can get the time course data is live cell imaging. But the live cell imaging, you can see only 2 molecules, or I think 2 molecules at one time. We cannot see that the whole dynamics or relationship of the dynamics from the live cell imaging. So that I think to the field, there's such a gap to understand the dynamics and between the of the molecular activity and the cell-fate regulation. We need a computational method, and we started to use that NLP. And NLP, by now you just using for the making a model. But I also think that it may be good for the integrating like live cell omics and other data as a NLP as a kind of intermediate tools. And also I hope that we can predict more like several diseases and aging and something like that, and also predict something good for the the biomarkers or something like that. And here is my laboratory. So the Kiwamu and Masato right now they are working very hard to making a model. And the former members definitely Hiroaki and Sawa. Actually, she was very good at handling TCGA data. And Hayate, he did a lot of MD simulation on the EGFR. They contributed a lot to our current project. And also we collaborate with the other members from Kyoto and Riken and Osaka University for the natural language processing and MD simulation. So this is my private research talk, but now I will talk about the general thing about Osaka and our institute. So maybe, I think, I don't know how many people have been to Osaka, and Osaka University is here, just next to Kyoto. And from Tokyo to Osaka, it's about 600 kilometers. So if you can, I think you can drive in one day if you want. And there is a castle. It's called Osaka Castle in there in the center of Osaka. The special thing in Osaka is local food. So that I have many visitors from Europe and America, and they really like this local food. I'm sorry, I didn't show ramen, but they like ramen a lot. But here is like pancake, but not sweet. It's like a meal pancake. And this is called octopus bowl. So there is an octopus inside. This is very popular in Osaka too, and other things. So that it's really, you can enjoy. Actually, our collaborator enjoyed this food very much. And this is about Osaka University. Osaka University was founded about 300 years ago as a medical school as their initial the foundation. And we have about 3,000 academic staff and 25,000 students and so on. And we have a School of Letters, Laws, and Medicine, Pharmaceutical Science, and Human Science, and Economics, and Dentistry, and Foreign Studies, Science, and Engineering, and Engineering Science. Oh, yeah, so that it's really big. Because of this big size, we always have the trouble to get the funding from the government, because we cannot afford this kind of huge number from just money from the government. So this is about our institute. So actually, even though I studied systems biology, my institute is basically for the protein sciences. So we have nice machines for the cryo-EM and beamline, X-ray beamline, and NMR, and so on. And we have a little bit of a small computer in our institute. And solve this kind of big, these days they like to solve a big protein structure. And our institute is founded in 1956, and solved the first cytochromes 450 enzyme structure by this people. And the most important thing these days is that we host PDB Japan. As you know, PDB is maintained by the England and America, and another is Japan. And PDB Japan is hosted by our institute. And because like AlphaFold and so on, recently we established a new center which is called ASPIRE, Advanced Data Science Center for the Protein Science. So this is a PI of the IPR. So we have many kinds, like protein chemistry, protein structure biology, and protein functions, and protein network biology, which I learned, and many other things like industry-funded laboratory and so on. So this is what we are doing for the Advanced Science Center on the protein research. This is on the making a new protein from protein structure, learning from the protein structures. Because we have a lot of PDB data. Why not making using such information to make a protein? And the good thing about our institute is that we can truly make a protein based on the prediction of the computation. So that right now we have the Paola Laurina. She's a visiting professor from OIST, southern part of Japan, and Professor Koga. He's really expert of this de novo design of the proteins. And right now we are trying to make the SARC kinase design to make a new inhibitor, and also the protein stability of that protein. So that we actually encourage a lot of collaboration with this center, so that I hope if you have time, you can visit us for discussion and seminars. So thanks for listening. Gracias.
So thank you very much for this very nice talk. We have time for some questions.
Thank you. It was really insightful. I liked it a lot. I have many questions, but I don't want to monopolize the whole thing. But regarding the patient-specific models, you mentioned that you were using at the molecular level, assuming that at the molecular levels, parameters would be the same for all patients. So this is indeed quite wise. Do you expect any effects of epigenetics?
Yeah, but I think the epigenetic information is we expect already in the transcriptome data, because the epigenetic change affect the transcription, so that we use the transcriptome data. So that I think epigenetic data is already there. Yeah, but and maybe further, modeling. For example, right now we use a bulk sequence data, but to estimate like a blood cancer, we need a single cell things. So that for that maybe we often use the attack sequence, attack-seq, because it's good to predict the transcription factor activity. So I think that will be that is possible, but will come later, because still it's difficult to integrate those things. So.
And do you think using metabolomics, because there is there are a number of papers now saying that there is a clear connection between some lipids, for example, with inflammation and cancer. Do you think this could be useful?
Yeah, I think so, definitely. Actually, I just focusing on the computational part today, but we also studied cellular senescence and aging, and we also took metabolome data of the aged cells and the non-aged cells, and we see that the dramatic metabolome changes, particularly the purine metabolism, and also TCA cycle. Somehow succinate was very much high in aged cells. And I think they generate reactive oxygen to make the cell more, aged, I think.
Okay. Just the last question. The simulations at the molecular level, this is something we were indeed discussing in the group to go for that. But because we are playing with microorganisms and we are playing at a large scale level, so I was wondering how does the approach scale with the number of proteins in your case?
Yeah, that is a problem. So, I show that even the easier computation is very difficult. But also, the problem in the structure of the protein, particularly for the signaling protein, they don't have a full-length structure. It's just a domain because they have a very, the intrinsic distal IDR region, which doesn't make a protein structure. But this is important for the binding of the other protein interaction. So I don't expect that we can solve this, structural biologists can solve all those signaling protein. But one of the things is, anyway, let's try the AlphaFold to see that, to estimate the full length and do this virtual, interaction simulation, for the whole signaling pathway and see how the mutation affects those things. Actually, I want to do that if somebody, that such a person who can, be interested in, I definitely want to collaborate with.
Okay, good.
Thank you for your question.
Thank you. Any other question?
Maybe I have one. In the model that you were using the publications for the last few years or whatever, how you deal with changes in nomenclature or changes in new discoveries, because papers sometimes different things are called the same thing.
At the end, I think we need a manual curation. Yeah, that is true. But it's still, I think this method reduces our manpower to spend the time to look at all papers, because we can, the extractor, because we searched with the PubMed ID or the gene ID, we can go back to the original paper, so we can see it if this is good or not. Yeah.
I have one question regarding the first part of the talk. So this molecular dynamic approach is very, very interesting because basically it's like a first principle approach to everything. But there seems to be a computational bottleneck there, right? Okay.
Very much.
Very much. Okay.
Yeah, or because of, I think just because of lacking of biological data, I think, because if the data is there, we can predict now, so that it's very hard. As I said, what we biologists can do is very much limited. So I think we need to use a different, approach to compensate, such a gap.
Then, of course, in the second part, this is extremely interesting because in biology there are so many papers. For a specific topic, there are hundreds, thousands of papers every year, so we cannot read them. So it's nice if the machine can read them, but it would be nice if in every experimental paper there would be a machine-readable description, unambiguous, right? Because then for this approach it would be excellent, because the problem is that we use natural language, which is somewhat ambiguous many times, right? But many people have been advocating for in every experimental paper to have a machine-readable description using some standard notation. But as far as I know, this is not happening, right?
I saw that it's I explained about the text model, and the text model says A binds to B. So I saw that we can extract such A and B relationship in the paper, and actually I by myself checked. There is any sentences describing A and A bind to B. Actually, no paper. It's written in more complicated way. And the text model is very good for the normalizing such a, the noisy sentences to make it in a more simple description. So it's also reduced that, how do I say, it can be more machine readable by using a text model.
For example, when I read experimental papers, it helps me the figures, because the figures are a better representation sometimes of the But now these LLMs can also incorporate images, right?
Yeah, yeah.
So are you thinking about also reading the images or interpreting that?
Yeah. My collaborator says it can be possible.
Okay, because I think it's probably a good reinforcement of this approach to incorporate the images.
Yeah. But what we use from PubMed is just abstract, because the abstract is free, but the content we have to pay. But I heard from someone from Sony that Sony is buying papers. Scientific papers, because they have a lot of money. So I wonder what they're gonna make for this method. Yeah, I'm really curious.
Okay. So, interesting.
Yeah, it's interesting.
But now we are forced to do open science, right?
Yeah.
So maybe, I don't know.
If you're Sony, maybe not.
Well, for the others.
So more questions?
Yeah, I have a, going back to the question about molecular dynamics. It's really very expensive to make a simulation in molecular dynamics. But I wonder if you know someone that are trying to solve the molecular dynamics problem using neural networks, as there are some examples in fluid dynamics, for example.
Yeah, actually, maybe there is someone as I said, these days somehow I'm in a committee of FUGAP, and actually, but my role is evaluating a biological the project which is FUGAP. But because of my role, sometimes I listen to a project related to the earthquake or the weather prediction, and they really use so many different methods. What you said, I think there is such a people in those projects, because I saw that it's really hierarchical simulation methods they use, and far more, the high standard and the biological simulation. So I think I'm sure that there might be. So that if you
in weather prediction, there are many works about to use deep learning for making predictions. In fact, we are working here with one institute, one office, our office, in order to create this. Molecular dynamics is one of example that consumes a lot of resources here, and because it can be used, maybe it's a good try to find a deep learning model that can do the same work as the molecular dynamics, but faster. Because in this case, you can link here with your model, with normal methodology. In this case.
I know someone who is indeed doing that. Using CESGA.
Today in the car we were talking. What percentage of CESGA uses of users are people doing molecular dynamics and structural chemistry in general?
Really, I don't know at this moment. Maybe Carlos, are you there? No. Last time that I saw these statistics, I think that 20% or around 20% of the total resources were devoted to only molecular dynamics.
20%.
It's a lot.
But it's something that is changing continuously, because depending on the research group that are using our system, sometimes we have more works from fluid dynamics, for example, and sometimes we have more from chemistry with molecular dynamics. The advantage of molecular dynamics is that they can use the GPUs very efficiently, and for this reason that we have a lot of works for using molecular dynamics here, because they can parallelize very well and they can scale very well. And usually the problems are very hard, because the challenge is to go to many seconds. I don't remember exactly the seconds that you have to simulate for making a binding or having a binding from a protein with a small molecule, but usually the people told me around seconds, because it's the time for the reaction sometimes. And simulated with molecular dynamics seconds is a lot of time.
And you were doing microsecond.
Microsecond right now, but we try to reduce the running time to 200 nanosecond and repeat more than the, because the variation in the structure is, we can see a lot of variation.
I have a second question about the papers, because there are a lot of papers with corrections after the publication, so someone that retried the paper because the data are wrong. It's not clear to me how you manage these rejections or these kind of things.
Yeah, actually we didn't check, but simply because we can see a title of the paper after the query, so that if the there is a erratum or interaction in front of the title, then we can know.
But I'm thinking in something like, for example, ChatGPT. You have there 1 petabyte of information without any curation. Do the job for it. So in this case, you have to analyze not only the information, not the retractions of information.
I understand. So that right now, what we are currently considering is that after log, we can get actually prediction is not so bad in some cases, but in the prediction it's not that good in some cases. So that we try to get the right, top 5 papers to, send it over again.
Okay.
Any questions? Yes.
Thank you. My background is in physics and it's always enriching hearing from other fields. I have a question. When you were showing the signal network, I noticed that you don't have an apoptosis pathway, and there's a relation between cancer proliferation and apoptosis. Wouldn't be that revealing?
Actually, we after that, the proliferation and the differentiation is our old study, and right now we are using the same cell line, and we are looking at the cellular senescence, cell aging, and also its relationship with the apoptosis. Because the cancer cell, we want the cancer cell to be dead after the radiation therapy or something. But in fact, they stay, but get aged. And people say, cancer scientists say that aged cell in the G1 is stops the growth, slow the growth. So thereby they can be resistant to the drug. So that and we found that the apoptosis and the senescence is kind of mutually exclusive in our experiment. So that and what we are trying to do is to reactivate the senescent cell to be more sensitive to the inhibitors. And then we also found that there is the oxygen reactive mitochondria is important to reactivate the senescent cell to be more active and to be killed by the drugs. Yeah. Thank you. Interesting question.
Okay. Next question. Okay. If not, we thank you again.